Everybody, now we're going to talk about the phylum Chordata. So in our previous lectures, we looked at, we've been going up the phylogenetic tree here in our Animalia, and we had just recently looked at the difference in bilateral animals can be separated between the protostomes and the deuterostomes, right? And so now we're looking at the groups that are part of deuterostomia, the clade deuterostomes. And so we shared similarities with the phylum Echinodermata, and that was the previous lecture, right? So if you need a review on the difference in the split between protostomes and deuterostomes, please go ahead and visit the Canvas page where there's a nice write-up on there for you. Okay, so again, we looked at the echinoderms. Now we're going to look at the phylum Chordata and see what are the characteristics that create this split. All right, so in the phylum Chordata, there are 4 key distinguishing innovations, or what we would just call the key characteristics. Right, so the nodal cord is this structure. It's a flexible fibrous tissue that surrounding fluid-filled cells. It's stiff, but it's flexible, and it provides skeletal support for all the primitive chordates. And what we're going to see, it also kind of provides that support to the dorsal hollow nerve cord, which would be like our spinal cord. And then later in the vertebrates, this will be a structure then that surrounds the spinal cord to provide protection. So, but in either case, it is a structure that's in between the digestive tract and the nerve cord, right? Let's see. What else do I want to say? Yeah, I'll leave it at that. The dorsal hollow nerve cord, then, this is the nerve cord where all our nerves branch off. It may have ganglia along there. This one is just showing a simple one. Okay, and what's important here is that in the chordates, it's on the dorsal side of the body or the back of the body, right? We saw on arthropods and the other protostomes that have a nerve cord, it was on the ventral side of the body. So that was a characteristic noticed early on as anatomists studied the different animals as well. The post-anal tail, then you can see, is a structure that extends beyond the anus. Okay, so aquatic chordates like fish use it for locomotion. In terrestrials, it can have a variety of functions. So many animals that have a tail use it for balance. You'll see in the Mammalia lecture, they use it for grasping. So some can use it, it's called a prehensile tail. They can use it to grasp and hang on in the trees. But in humans and in the other great apes, it's absent. We don't have it that extends beyond the body. What we see is under the skin, we do have the tailbone, which are called the coccyx bones. So in most other phyla, the anus is the terminal end of the animal, but in chordates, we actually have the tail that extends beyond it. And then the endostyle is along the pharynx. It's a groove on the floor that has mucus and cilia, and it helps to trap food. And so again, we'll see that in our embryonic development, this will be the case, and in some of the organisms in the phylum Chordata. In some, though, the embryonic development, as it progresses, we lose it in the adult body, and it becomes the thyroid gland, actually evolved from that, the gland that sits right underneath our Adam's apple there. And then the other one, there are the pharyngeal slits. So the pharyngeal slits are close to the mouth, and they open to the outside, right? And it allows water to enter through the mouth and then exit through the pharyngeal slits so that it doesn't go down into the digestive tract. In early chordates, it functioned as a filter feeding device. In others, it's a gill, it becomes the gills for gas exchange, right? And then in terrestrials, it gets modified to other purposes, like the tubes in your ear, the eustachian tubes. It also forms some structures in the neck, but eustachian tubes kind of come down there as well. And it's also found in echinoderms too, so that shows, again, a little bit of relationship with the echinoderms. Okay, other characteristics that are typical to most chordates, they might not all have them, but there's if they have a skeleton, it's an endoskeleton, and it might be made out of cartilage or bone or both. They have a closed circulatory system, a ventral heart, so it's on more of the front side of the body, and there is segmentation. And it's so specialized that the basic segmentation of the body plan, it might be difficult to see, but we do get hints of it in like vertebrates, again, repeating body muscles. So along our back, we have repeating body muscles. We have the bones of the vertebrae, and then also the nerves and the ganglia, right? So that's the indication of segmentation. So remember, segmentation evolved 3 times in Animalia, not from descent. Okay, so it developed in the annelids, right? It developed in the arthropods, and then it also evolved in the chordates, especially the vertebrates. Okay, so within the phylum Chordata, we have 3 subphyla. So remember that traditional classification is king, phylum, and then we go to class. But every once in a while, when they haven't worked out the organisms within a different classification, they throw in a subphylum in there. So we have 3 subphyla: the Urochordates or Urochordata, the Cephalochordata, and then of course the vertebrates. The vertebrates are the most diverse out of these 3, and again, by species, by their body structure, and they also occupy all biomes of the earth. The first 2 subphyla are much more simple. Well, let me show you what these animals are. So the first one in the Urochordata are the tunicates. Tunicates are these gel-like organisms, and they, as adults, they attach to rocks and things on the sea floor, so they're sessile. Okay, and this is the inside of what a tunicate body looks like. There's actually about 3,000 species described. So the only thing that they retain from those characteristics of chordates in the adult body are the pharyngeal slits. And so it becomes, sometimes it's referred to as the pharyngeal basket, but again, still, it opens up, water comes in, it can filter, and water can exit, and that water doesn't enter the digestive tract, but the food particles do. Okay, so they're really a filter feeder. It's their larvae that are bilaterally symmetrical, and they have all the parts. They have the dorsal hollow nerve cord, they have the notochord, they have the pharyngeal slits, which function more as gills in the larval stage. Okay, they have the post-anal tail, and there is an endostyle in there as well. Okay, it's the whole animal then is really just enclosed in a tunic, and so that's where they get the name tunicate. It's kind of a gel-like body substance. There is a very rudimentary circulatory system, and there's a very simple nerve system. They do have that dorsal hollow nerve cord. Most of them are also hermaphroditic. So here's just a few other pictures. You can just see how delicate, how thin they are. Right? They mainly get their gas exchange by diffusion. Okay, so that's all you need to know for the urochordates. The subphylum Cephalochordata are the lancelets. These are little organisms called lancelets. They look fish-like, but they're not a fish because fish are vertebrates. There's only about 26 species, and all of them are marine, and they're filter feeders. So they have the pharyngeal slits, and again, they bring in water through the mouth, the endostyle in there. The water is going to exit the pharyngeal slits while the food heads down into the digestive system. Gas exchange, though, there's no respiratory system because gas exchange can occur across all the body surface. They're just like a millimeter or 2 long. They're very, very small animals. So they are sessile. They tend to burrow down into the sand and just filter feed. But again, you can see these arrangement of muscles in like a V going across both sides of the body. That's called a chevron. And they can contract each side of the body, the muscles at different sides, and that forms a swimming movement. So they do have the ability to swim at least short distances. And again, that might get them away from predators that are come along, or predators that are suck the sand and get the organisms out of the sand. Right, so the swimming motion is really characteristic of fish, like I said, and it might have been the precursors to our modern fish. Okay, and so when we compare these, how can these two organisms be related to humans, right? Again, along with the structural similarities of the nodal cord, the dorsal hollow nerve cord, the postanal tail, etc., we have compared the small unit ribosomal RNA. And so again, here it is between the human and a lancelet. We see a lot more similarities between those than we would see between a human and a mollusk, or even the lancelet and the mollusk, or even the lancelet and the mosquito. So again, molecularly, when we see more similarities in the sequence of DNA, RNA, that shows a common ancestor, a more recent common ancestor. The more time between divisions in evolution, the more differences, the more mutations can develop between the different species, and the more distantly related they are. Okay. I took a little note in the urochordates, those tunicates. There's a species called Siona. About 80% of its genes are found in vertebrates. So that's pretty good. So the body form of this urochordate might actually just be the derived form. Okay, so it might have been more closely, its ancestors might have looked more like the larval stage in the past, and then the adult body in this species, in this modern grouping, became the derived form. All right, so that brings us to the subphylum Vertebrata. And we're going to include a little outgrouping of the vertebrates. They're closely related. They don't they don't fit in the other two groups. They're more closely related to the vertebrates, but the one thing they lack is the vertebrae, right? So let me show you the characteristics of the vertebrates first, which is what we see here. Again, the vertebral column is very important, but also a cranium, right? So this is a hard covering to protect the brain, to protect the anterior side of the nerve cord is where the brain develops. So we see first this cranium develop, and then we see over time that notochord again becoming part of the vertebral column, right? So it just continues this trend of cephalization, getting more extreme cephalization. Again, there's going to be an endoskeleton in all the vertebrates. It can be bone or it can be cartilage. Sometimes it's a little bit of both. We have a mixture of bone and cartilage in our body. Our bones are capped with cartilage. It's very important to prevent our bones from knocking on one another. Okay. The neural crest cells. So we saw some of this when we looked at the nervous system of animals in the physiology section. And so I'll show you a picture. Well, I'll just do it right now. The neural crest cells. Remember that the endoderm of the three germ layers forms a flat tissue, but then there are these neural crest cells, these little cells along a ridge that get attracted to one another. They send cell signaling, and it causes them to fold and want to meet with each other at the middle. And when they do that, that causes a get my laser on, a space in the middle. So this is the hollow nerve cord that's developing. Okay, and down here you can see where the nodal cord is, right? And that is going to differentiate into bone. And some of the nerve cord is retained in discs that are between our vertebrae. So you might have heard somebody has a slip disc. That's the cushion between our vertebrae. That's derived from the notochord. Okay, so this long tube is what forms our spinal cord at one end, and at the anterior end it forms our brain. Okay, that's where that develops from. Okay, we have duplicated Hox gene clusters. All invertebrates and non-vertebrate chordates have at least one. And again, the Hox genes are the sequence of genes that create our body plan. So they get turned on at particular points during embryonic development, so that these are what provide us with maybe that sequence of the vertebrae. Or we all have 4 limbs that stick out. You'll even see in fish, they have pectoral fins and they have 2 pelvic fins that stick out, right? So that's the Hox gene plans that we have. Okay. Pronounced cephalization, so a bigger head, bigger brain developing compared to the rest of the organisms we've studied so far. And now we have nerves branching off the brainstem. Okay. And again, paired appendages, most have that, and then some will lose them. I gave you an example of the dolphin that's lost the lower appendages. They don't need it in their environment, but they have the genes for it, because once in a while we find that anomaly where we see a dolphin with lower pectoral fins and they shouldn't have them, or lower pelvic fins, I'm sorry, and they shouldn't have them. Okay. All right, so let's go through the phylogenetic tree and look at each of these groups and their characteristics. So again, the outgroup is this Myxini. So we always talk about them along with vertebrates, but again, they're not quite vertebrates. They do have a cranium, but they don't have vertebrae. The notochord is their only support. Okay, but the groups that are in there they're called hagfish. They live in the deep ocean. I encourage you to click on this link. This shows you some researcher here at Scripps Institution of Oceanography here in La Jolla, who studies hagfish, and he shows you their defense mechanism. They have glands along the sides of their body that produce a slime that can deter predators. And once the slime hits the ocean water, it just grows and becomes this big mass of slime that can get in the nose and the eyes of predators, and they want to get away with it. So in this video, he shows this happening. It's pretty interesting to see. And if they get slime on themselves, they actually have the ability to sneeze to get the slime out of their own nostrils, right? The other thing that links them to the vertebrates is now the molecular evidence. So it does show that they did evolve from vertebrates. So they might have just been, again, sometimes some more recently evolved organisms go back to the ancestral trait. And again, these are deep ocean organisms, so they may not, the bones may not be the best made for that compression with the pressure coming down from the ocean. So it might be better to have the notochord. Who knows? Okay. This brings us then what we would call the fish. All right, but not all fish come from the same class. Sorry, I almost forgot what we were talking about here. In the same class, but so fish are swimming vertebrates. Many of them have a jaw, but not all of them have this hinged jaw that I was telling you before. So this is of course, this is a deer, but it's on a hinge, so it can open and close. Right. If it's a fish that does not have a jaw, in general, we refer to them as agnathans. Again, it's just like a clade. It's just like a bunch of individuals that share the same trait that's probably from descent. We refer to them as gnathostomes if they do have a jaw. And again, they'll have these additional Hox clusters which help to form this really robust jaw. Okay, well, let's look at it on the phylogenetic tree. So now we get a split and we see there are going to be jawless vertebrates compared to those that have jaws. So from there, we're going to start with chondrichthyes, which are sharks, appropriately jaws, right? But then the other fish, Actinopterygii, Actinista, and Dipnoi, they all have jaws, as well as we get onto the terrestrial environment, amphibia, mammalia, and reptilia. Okay, so let's first look at the jawless fish, Cephalospidomorphi. These are the lampreys. So they don't have a jaw, they have a circular disc for their mouth. Okay, and what they do is they have these bony projections and they can use that to attach onto the side of a fish. This one's dead, so they probably did that to get a picture. But they can attach to live fish, but they do end up killing them. And they just rasp away at their flesh with this tongue, similar to a radula that we saw in the mollusks. And they just rasp away at it. So again, they lack the hinged jaw. They also lack true appendages, but they do possess a notochord and also a rudimentary vertebral column. So it's a cartilaginous rod now that surrounds the notochord itself. And so we're starting to see that getting closer and closer to a true vertebrate. But it is considered a vertebral column. So it's one of the most primitive groups of vertebrates. They're found in both marine and freshwater. They can go back and forth. They're a big problem in the Great Lakes around Michigan because they've flourished in the Great Lakes and they really attack a lot of the game fish that are in there. So again, this is considered like an ectoparasite. They are parasitic as adults. So let's look at the evolution of the jaw. Then the jaw came along, right? And the jaw developed from what are called pharyngeal arches. So even this lamprey, it's hard to see, but they have gills. All right, actually, they're right there. So they have gills along there, and those openings open to the outside. Okay, and so they have, in between their gills, they have these structures called arches that kind of hold open the skin so that water can flow into the gills much more easily. So those are called gill arches. And we can genetically identify those, right? So what happened is, here's an extinct early jawfish called a placoderm. They were really heavily armored fish, really thick armor on the outside of their body. But we can sequence the DNA and see that gill arches 1 and 2 were lost, and then gill arch 3 is the one that's modified to become the hinged jaw. Again, just comparing DNA with those that have a full complement of gill arches. Okay? And then the cartilaginous fish developed. Right, here are living forms of fish, and now we see that gill arch 4 also became modified into a heavier jaw for more efficient grabbing of prey. Okay, so this is gill arch 2 and 3 now become the jaw. And this happened in about the late Silurian, about 417 million years ago is when the jaw started to develop. Okay, and so this really shows descent with modification. Remember, we talked about that with Darwin's ideas, descent with modification. Body parts just don't suddenly appear in the fossil record. It just isn't suddenly that, oh, fish have jaws now, right? We actually see a little stepwise between no jaws, between a modified version of a jaw, and then a jaw, you know, the full jaw that we see now. And now we can use molecular evidence that shows that the jaw probably could not have evolved without these gill arch structures. So that set the blueprint to allow this modification to occur. Okay, so again, a hinged jaw is a significant evolutionary development because you can grab prey more firmly, and so this could probably increase the rate of capture and then even help to attack larger prey, and then increasing the food supply. And of course, you're going to be successful if you can do that. So let's look at our hinged jaw. So again, that jaws would be here, and then our first group then would be the class Chondrichthyes, right? And those are our cartilaginous fish, and these include sharks, skates, and rays. The cartilaginous skeleton, endoskeleton, is a derived character, not the ancestral character. So even though our bones develop first from cartilage and then into bones, so when you're developing as a fetus, your skeleton develops as cartilage and then becomes bone. Okay, you would think then that the ancestral trait is the cartilage. And what we see with sharks and skates and rays, you think, well, they must be the ancestral group, but they're not. The molecular studies show that they are descendant from bony fish, and then they went back to the ancestral trait of the cartilaginous skeleton. So therefore, it is a derived characteristic. So in the class Chondrichthyes, there is a change in the developmental sequence that prevents the ossification of the bone. Okay, cartilage ossifies and becomes bone. So they have a gene then that stops that. And it just gives so much more flexibility to the skeleton and their fins and makes them really powerful swimmers. The other thing about them is, and I'm going to show you some pictures, but the sharks were among the earliest fish to develop teeth. But their teeth are a little different than our teeth. They're developed from scales that actually also contain dentin and enamel, and these are two compounds that are found in our teeth. But they're actually derived from scales. Ours are derived from bone. Theirs actually break off really easily because they're not set into the jaw. So sharks have the ability to continuously replace their teeth row by row. They kind of rotate forward. They can have up to 20 rows. So here you can see powerful swimmers. They're going to have muscles on either side of the body, left and right side, so that causes their tail to swish back and forth laterally. Dolphins, remember, they evolved from terrestrial animals. So in terrestrial animals, on your legs, you have muscles in front, you have muscles in back. So when they adopted the posture to swim in the water and lost legs, but those muscles then became the top portion of their tail and the bottom portion, dolphins and whales swim this way, right? Again, because they're using the traits from their more close ancestors. Fish, their close ancestors you saw in the lancelets had muscles on left and right sides, so they're going to swim this way. Okay, but again, cartilaginous skeleton makes it very flexible, so you get really big, powerful movements in that tail. And sharks can be very fast. They can't sustain this fast swimming for a long period of time, but they do it in bursts so they can grab their prey and get out of there. Okay. The rays, what happened with them is the sides of their bodies flattened out. So they actually swim, instead of the tail being the powerful muscle, they actually swim with the sides of their bodies, right? And their mouth is on the ventral side, right? And eyes up on the dorsal side. So this group Chondrichthyes, they have internal fertilization. Right, so the males have, let me see this picture here. This is a female. So males would have these extra projections coming out called claspers. I wonder if I can just real quickly stop sharing this. Let me share my, I have to share, I can't find it. Nope. I'm going to pause this so I can find this for you. Here we go. So I should be sharing this screen. These are the claspers on a male shark. So these are what sharks use to grasp onto the female and hold on to her for mating to occur. Right. And so here, We can see this a little bit better. Nope, let me go back to the other pictures. Let me do that. Right, so you might be able to see on here. So the claspers, these are what the male sharks are using to hold on to the female in order to deposit sperm into her body. Okay, so let me screen share again. Let me get my other, wait, let me stop share. We'll start over again. Hold on, maybe I should pause. It looks like I lost my place on the PowerPoint, but I'll get it back. Okay, ready to screen share. You can fast forward through this, sorry guys. Whoops, trying to. There we go, all right. So yeah, so this is a female, so males would have those projections, and that's what helps them with internal fertilization. Okay, and so the fertilization can happen internally, but then the female could lay the eggs like this. This is sometimes called a shark purse. It's what they refer to for eggs that have been laid, and then the embryo develops outside of the body, but inside of this egg case. Right, and you can see the embryo inside of there. Those are cartilaginous fish that are oviparous, right? They lay the eggs. Some produce an egg like this where the embryo develops, but they retain it into their body, and that's called ovoviviparous. Ovoviviparous, sorry. So it's retained in the female. There's no placenta. There are very few species of sharks that are actually viviparous. And so they produce a rudimentary placenta. It's not like the mammals, but it develops inside, again, a uterus, not very similar to mammals. But that's what nourishes the embryo growing inside of there. And they'll actually give live birth. And there are some shark species that are so aggressive that as the pups are developing in the uterus, as they start to get older, they're not born yet, they'll actually eat each other in the uterus. There's competition within the uterus. And then when they give birth, when the mothers give birth, the pups have to swim away so fast because the mothers might turn around, they recognize that as prey, and they might try to eat their own pups. So they have to disperse away from mom. Talk about wonderful parental care. So that's just a few species of pretty aggressive sharks. Okay. So the next group are the bony fish. So there's actually 3 living classes of bony fish. So the difference now, the endoskeleton is made out of bone, okay, but the skeleton is a little bit different in each of these 3. And there's other 3 different, there are different characteristics for each of these. But what's common between these 3 and the chondrichthyes, again, the bony skeleton. Also, notice, maybe we can see in the shark picture here, yeah, the shark also, their gills are open to the external environment. They're just open outside of there. And so remember, the gills are just a structure that's full of blood vessels. And so if they get bit or damaged, then they could bleed out through their gills. So the bony fish have what's called an operculum. And that's just a flap that covers the gills. And so the gills are protected, but the flap can open up to allow water to move out. So again, water comes in the mouth and moves out the gills, and the gill structures now can get the oxygen, right? The other thing we'll see is the swim bladder for buoyancy. So that's the structure inside here. So this is a swim bladder, it's gas filled, and again, that can help different fish hang in different areas of the water column. So some like to be at the surface and they can sink down a little bit. Some are very deep water fish and they need to be weighted down a little bit more, so they would have less air in there. We'll see that there's one fish that's their swim bladder is oil filled, and so that's going to allow them really to stay at the deep depths of the ocean. Okay, most species of these fish reproduce through external fertilization. the eggs do tend to sink, and so many fish actually spawn or release their egg and sperm in shallow waters. And so coastal areas are really important for the health of the deep ocean fish because they're coming up to the coast to lay their eggs. And when we pollute the coastal areas, we have all that pollution that can kill the eggs, and that can cause a population crash of the deep ocean fish. So it's really important that we have a lot of, that's why we have a lot of protection, like construction sites that are near the coast. They have to be very careful about not only just pollution, but all the sand and dirt that might get into the waterways. So even if you're pretty well inland, we do have waterways that bring water to the oceans. And so we have to make sure that we're clean all the way. Not just fish, but even some of the arthropods, mollusks, they all reproduce in the shallow areas. So very important. So again, let me show you the three groups here. We got Actinopterygii, Actinistia, and Dipnoi. Okay, so these are all bony fish. And we're going to show what their characteristics are, separating each one. Okay, so the first one we'll look at, you see, so these are all jawed fishes because the jaws are down here. And then you got chondrichthyes, and now we've got a bony skeleton. That would be the trait here. Okay, so but what separates Actinopterygii? These are called ray fin fish. So in their fins, they have those really tiny, thin, bony, flexible rays, and that helps them. They can move their fin as a whole. Mostly it's used for balance. It can be used for display, like this lionfish. They can be used, modified for camouflage, like this is a dragonfish that hides in the kelp, right? Wide variety of fish. I mean, I've only got a few pictures here, but if you recall from the vertebrate skeletal lab activity that we did, you got to see those bony rays. So you still have access to all that on Canvas. So if you want to go back and look at those pictures, you can. All right, so again, lionfish, these guys are really territorial, and when you're diving and you get into their territory, they'll come up and they'll fan their rays, their fins at you. But they have poison glands, so you want to be careful. You can't get too close to them. Right, and again, here his fins are mimicking the kelp behind it, and that's just through natural selection. So that fish that had fins that kind of mimic the kelp, a predator probably bypassed it. Okay. Actinistia is the next one. These are the coelacanths. And so what they do is they have a special joint in the skull that gives them a much more powerful bite, and their fin bladders are filled with oil. Right, so these are deep water fish, very deep water, and so they need the oil in there to help hold them down, and so they don't float up, right? But that's why one reason why we thought they had been extinct. They're hard to come by, but we realized no, they're not extinct. They're actually off, I think, off Africa and Indonesian waters. That's it. They were really successful in the Devonian. So we have a lot of fossils of coelacanths, and they seem to be abundant. And that's why we thought we thought they died at 65 million years ago at the time of the dinosaurs. So, but the other thing, it's a lobe fin fish. So look at how much thicker their fins are. They have stronger bony structures in there. There's actually skeletal projections coming from the pectoral areas and the pelvic areas back here. And again, so that sets the stage for moving onto land, right? The ancestors of these coelacanths of Actinistia were probably lungfish, but then they lost the lungs and returned to the seas. But this other group, Dipnoi, class Dipnoi, they have the lungs. So they are also lobe fin fish. They have the structures in their fins, but they have the lungs. Okay, but that allows them, when a pond dries out, they can dig and burrow and go dormant until the next rainy season. So they use those lobe fins in order to do that. They can also transverse over dried out areas to get to another pond, right? So they can actually go out over land and use their lung to kind of breathe air, right? So they need both gills and the lungs. They do tend to live in oxygen poor fresh water, so both of them help them to get the air that they need. And they'll actually drown if they're not able to come to the surface and get the air for their lungs. So kind of interesting. So there are morphological, like in the morphological, again, the physical traits, like in the fins, but also molecular similarities of coelacanths and these lungfish, dipnoi, to primitive terrestrial animals. So again, suggesting that their lobe fin ancestors probably gave rise to the coelacanths and these lungfish and tetrapods. Okay, so that's still being worked out. Okay, amphibians, the next ones. The next class, amphibia, right? So again, back to our lobe fin fish, we believe they were probably the ancestors that were able to come on land. Again, ponds would dry out, and so these fish would spend some time on land, little short periods of time. So that gave rise to the tetrapods, right? Tetra means 4 feet. So that's what arises after, that's what you need to walk around on land, okay?